Have you ever just stopped and wondered what makes you, you? You know, what decides the color of your eyes, or why you have your dad's nose? Well, the answer is hiding inside this incredible, elegant molecule. It's the instruction manual for building a life. And today, we are diving deep into that code, the code of life itself, DNA. I mean, really think about that for a second. How can one single type of molecule write the entire story of you? And not just you, but every single living thing on this planet, from a tiny little bacterium to a massive blue whale. It's all written in the same language. So how does this microscopic thread hold the blueprint for your whole existence? Let's find out.
Okay, so that's the official textbook definition, and it's a mouthful, but it's also incredibly powerful. The best way to think about it is this. DNA isn't just a part of you, it's the fundamental recipe for you. It's the master blueprint that's been passed down for generations, and it has every single instruction needed to build you from just one cell and keep you running every single second of the day.
So to really get our heads around this, here's our game plan. First, we're going to look at the beautiful structure of the blueprint itself. Then, we'll see how that code actually gets turned into action. After that, we'll talk about the massive project to read the entire human instruction book, explore a really shocking discovery they made, and finally, look at what all this means for our future. All right, let's start with the basics. The actual shape of DNA. Its structure is probably one of the most famous images in all of science, and for a really good reason. The way it's built is the key to absolutely everything it does.
The fancy term is a double helix. You've got double because there are two long strands, and helix because they're twisted together. But honestly, the best way to picture it is just a twisted ladder. And this shape isn't just pretty, it's a genius way to pack an enormous amount of information into a teeny tiny space.
So if DNA is a ladder, what are the rungs made of? Well, they're made from just 4 molecules that make up the entire genetic alphabet: A, G, C, and T. That's it. And they follow one simple, absolutely crucial rule. A always pairs up with T, and C always pairs up with G. This perfect pairing is what holds the two sides of the ladder together, and it's the secret to making flawless copies of the code. Okay, so we've got this amazing blueprint, this super stable stored code. But, you know, a blueprint just sitting in a drawer doesn't actually build a house, right? So how does the cell read the DNA and turn it into all the stuff that makes us alive? This process is so fundamental to biology, it's called the central dogma. So the central dogma, it's really all about a one-way flow of information. It starts with the master blueprint, the DNA, that gets turned into a temporary message, which is RNA, and then that message is used to build the final products, the proteins. Information flows out from the DNA, but it doesn't flow back in. This is the basic operating system for pretty much all life on Earth. The whole thing happens in a few key steps. First, you've got replication. That's just DNA making a perfect copy of itself. But for the daily work of building things, the cell uses 2 other steps: transcription and translation. And honestly, the best way to understand this is with a simple analogy. Let's just imagine you have this priceless, one-of-a-kind family recipe book. It's been handed down for generations, and inside is the secret recipe for the most amazing cake in the world. You want to bake that cake, but there is no way you're taking that precious book into the messy kitchen. And that is exactly how your cells treat their DNA. The DNA is that master recipe book. It stays locked away for safekeeping inside the cell's nucleus. It's just too valuable to risk getting damaged, because it's the original, master copy of every instruction your body has. So what do? Simple. You carefully open the book and you copy or transcribe the cake recipe onto a small disposable note card. That little note card? That's your messenger RNA, or mRNA. It's a temporary mobile copy of just one recipe that can safely leave the library and travel out into the main part of the cell.
And this brings us to the grand finale. You take that note card to your kitchen, which in the cell is a little structure called a ribosome. And there, you translate the instructions on the note card to build the final product, the cake. In the cell, that cake is a protein. And proteins are the little machines, the workers, the building blocks that do basically all the work.
Okay, so now we get how one recipe or one gene works. Now imagine trying to read the entire recipe book from cover to cover. Well, that was the absolutely mind-blowing goal of the Human Genome Project, to read every single letter of our genetic code.
And the task was just monumental. The human genome isn't a short story, it's an epic. We're talking about 3 billion pairs of those A, T, C, and G letters. If you were to type all of that out, it would fill a stack of books 200 feet high. That's the staggering amount of information packed inside almost every single one of your cells.
This massive global project took more than a decade, but one of the most game-changing moments happened in 1996. Scientists from all over the world agreed to something called the Bermuda Principles. They promised to release all their data, publicly, for free, within 24 hours. That spirit of open collaboration, of sharing instead of competing, just supercharged the whole project, leading to the first complete draft in 2003. It was amazing. So reading the entire Book of Life was an unbelievable achievement. But what science has found inside its pages, well, it was maybe the biggest biological surprise of the century. It completely flipped our understanding of what a genome even is. See, everyone thought that the genome would just be packed with genes, those protein recipes we talked about. But the reality was shocking. Only about 2% of our 3 billion letters actually make up genes that code for proteins. 2%. So that immediately raised a huge question. What on earth is the other 98% of our DNA even doing?
At first, nobody really had a good answer. The main theory was that this huge, non-coding part of our DNA was just evolutionary leftovers, useless genetic clutter that we'd picked up over millions of years. It even got a really unflattering nickname, junk DNA.
Oh, but we now know that could not be further from the truth. That so-called junk DNA isn't junk at all. It's the genome's sophisticated operating system. It's full of switches and dials that control when genes turn on, where they turn on, and how much they turn on. It's what makes a brain cell different from a skin cell, even though they have the exact same DNA. It's the director of the whole show.
And this deeper understanding brings us right here, to today. We can now read our own genetic code fairly easily and cheaply. And this new power is incredible, but it also brings up some really important personal and ethical questions that, honestly, we all need to be thinking about.
Okay, one of the most important ideas to get is the difference between predisposition and determinism. For most diseases, your genes don't give you a diagnosis, they give you a probability. A genetic predisposition means you have an increased risk, but it is absolutely not a guarantee. And this is so important because it means your lifestyle and your environment still play a huge role in your health. Your genes are not your destiny.
Now, as this kind of genetic information gets easier to access, protecting it becomes absolutely critical. In the United States, there's a really important law called the Genetic Information Non-Discrimination Act, or GINA. It was passed to make it illegal for health insurance companies or employers to use your genetic information against you to deny you a job or coverage. It's a legal shield to make sure the amazing promise of genomics doesn't come at the cost of our basic rights.
And that leads us to the ultimate question. For the first time in the entire history of life on this planet, a species, us, has learned to read its own source code. And with new technologies, we're now learning how to edit that code. This power could cure horrible genetic diseases, but it also opens up a whole universe of tricky ethical questions. We have the instruction manual in our hands. The challenge for us, and for everyone who comes after us, is to find the wisdom to use it well.